Proteins are the most versatile of biological macromolecules. Much of it is owed to the variation in shapes that they take. Not only do they function as an energy source, but also for cell structures, cell signaling, hormones, immune response, movement, and enzyme activity. Proteins are split into 7 types. First are enzymes, which can either help build or break down molecules in the body. Amylase, for example, helps to break down starches into glucose molecules. Transport molecules like hemoglobin on blood help to deliver oxygen through the body. Actin and myosin are both structural and contractile proteins used for both cellular structure as well as muscular movement within the body. Some proteins, like antibodies, help fight off disease. Proteins like carbs and lipids also contain energy used to make ATP. Finally, some proteins like insulin help to regulate bodily processes. Unfortunately, protein structure and function can change in the presence of higher temperatures or changes in pH. This can cause denaturization, or changing the shape of a protein. Sometimes it's reversible, sometimes it's not. So proteins are made up of monomers called amino acids. Amino acids are the monomers that make up these proteins. They're composed of 4 parts: an amino group, NH2, a hydrogen, a carboxyl, either a COOH or COO negative, the ion form group, and an R side chain. If you remember from the last chapter, R just means carbon group. There are 20 amino acids in existence. All of them have similar structure except for the R side chain and whether or not the carboxyl group is ionized or non-ionized. So in this case, this picture on the left is a non-ionized amino acid. All life needs all 20 amino acids. And while most amino acids are produced in our cells, some like isoleucine, leucine, and cysteine are called essential amino acids. We retrieve them from our diets. So amino acids form chains called by polymerization. Polymerization happens by a peptide bond, which is a chemical bond formed by the link of a carboxyl group on one amino acid to the amino group of another, creating a carbon-nitrogen bond. So when 2 amino acids come in contact during a condensation reaction, 2 hydrogens from the amino group join the oxygen from the carboxyl group, and that causes them to be formed together. Since nitrogen and carbon lack a bond, they are bonded together, resulting in 2 amino acids joining. A chain of amino acids linked by peptide bonds is called a polypeptide. Polypeptides that contain fewer than 50 amino acids are called oligopeptides. Polypeptides containing more than 50 amino acids are called proteins. Proteins are really complex, and they have 4 main structures involved. And I want you to think of these as like the human body. We are functionally made up of cells. Cells make tissues. Tissues make organs. Organs make organ systems. And the organ systems make us. So we're going to start at the very basic structure of proteins, and that is the sequence of amino acids. So the shape of a protein is directly linked to its function, and thereby any change to that protein will either alter its function or make it non-functional altogether. Proteins are the overall molecule, but its structure can be defined by 4 stages: primary, secondary, tertiary, and quaternary. Think of these as hierarchical terms from the human body. So our structure, our primary structure is our cells. Our secondary structure is our tissues. Our tertiary structure is our organs. Our quaternary structure is our organ systems. So if you think of that comparison, we'll start with the primary structure of proteins, which is an amino acid. So this is the bovine serum insulin. The primary structure is literally just a chain of amino acids that make it work. If you change any part of that amino acid, it can cause the protein to be non-functional. Probably one of the coolest studies ever done on amino acid changes on a species. Take a look at a beach mouse. In Florida and Alabama, there's forest mice, similar to ones we have here in Kentucky, but there's a strain called a beach mouse, or a subspecies. And this beach mouse, obviously it blends in really well in sandy environments. And all of this is all thanks to a simple change in one amino acid. And that change in that amino acid is just enough to lighten the fur of this particular mouse. Unfortunately, beach mice are endangered. So let's take a look at the secondary and tertiary structure of proteins. The secondary structure of proteins has to do with how polypeptide chains fold with each other. The two most common folds are called an alpha helix and beta pleated sheets. Alpha helix, beta pleated sheets. Hydrogen bonds between the hydrogen of an amino group on one end binds to a carbonyl group on another end of a amino acid chain. Tertiary structures begin to form more three-dimensional shapes. These are formed by R groups interacting with each other, causing unique bends, ionic bonds. That's an ionic bond between two R chains, hydrophobic interactions between two nonpolar amino groups, hydrogen bonds similar to secondary structure, and finally disulfide bridges, which occur uniquely with just cysteine amino acids at different intervals of the polypeptide. And again, all of this contributes to the overall shape of the protein. Finally, the quaternary structure of proteins. Sometimes polypeptide chains made from the aforementioned protein structures interact to form a quaternary structure. So think of these as several tertiary structures binding together. So here's one tertiary structure and here's another one chemically bound together. So as a review, the shape of a protein dictates its function. Any change to the shape will drastically change its function or denature it. Temperature changes, pH changes, exposure to certain chemicals could cause a change in the shape. In some cases, a protein that is denatured is reversible provided that the change agent is removed. Sometimes it's irreversible. Albumin, for example, denatures while cooking, causing liquid egg white to become solid. Even when removed from heat, the whites of the egg will not go back to being liquid because you have denatured the albumin protein. This next group of biological macromolecules is known as the nucleic acids. Nucleic acids carry a cell's genetic information and information on how a cell should function. Two forms of nucleic acids exist, RNA and DNA. They only differ structurally by the addition of the hydroxyl group on the second carbon on RNA. Take a look here. The only difference between these two structurally, two hydroxyl groups for RNA, one hydroxyl group for DNA, and a hydrogen on this second carbon here. There are 5 nucleotide bases. 3 of them are pyrimidines, thymine, which is only found in DNA, uracil, which is only found in RNA, and cytosine, which is found in both. 2 of the bases are called purines, and those examples are adenine and guanine. Those are found in both. All of the purines form double rings. All the pyrimidines form single rings. The nucleotide base attaches to the first carbon of the deoxyribose sugar or ribose sugar, depending on the molecule that you're looking at. Since DNA is double-stranded, adenine and thymine pair up via 2 hydrogen bonds, and cytosine and guanine pair up via 3 hydrogen bonds. And we're going to learn more about this when we look at the structure and function of DNA. But just the basic structure of DNA, it's basically described as a double helix. When nucleotides polymerize, the hydroxyl group on the 3' carbon, this is 1, 2', 3', 4', 5' carbon, so the hydroxyl group here is then going to bond to the phosphate group on the 5' carbon of another nucleotide. When this happens, Dehydration reaction occurs, water is removed, it forces a bond between phosphorus and oxygen, creating a link. These are called phosphodiester bonds. And these phosphodiester bonds form this backbone that you see right here. These molecules are held together by hydrogen bonds. Hydrogen bonds are weak, they can be easily broken, but also easily formed back up. RNA's main job is the production of proteins under the direction of DNA. Again, we will look at this stuff later on this semester. So we are finished with this chapter, and now that we've learned about the biological macromolecules, we'll see how they all come together to make cells.